In this video, we will discuss about their AND logic gate efficiency on different cell lines, how they design their AND logic gate and how they optimize their AND logic gate to give only the specific signals and at the same time prevent non-specific signals. So the designing of this logic gate begins where they distribute the initiator DNA into two splits. One is designated to one biomarkers and another designated to other biomarkers, and this gives to a structure or to a junction which they call as three-way junction. As we can say, it can go in three directions: this direction, and this directions. So they wanted to find out what's the efficient length of this three-way junction can be used to give only the specific signals and prevent non-specific signals. So they tested three different nucleotide lengths. There is 4, 6 and 8 nucleotide. And also when these two biomarkers are close to each other, then only this junction will form. So if one biomarker is present on one cell and another on another cells, then these junctions won't form. And this is what their goal, to make this hybridization chain reaction only when both the biomarkers are present on the same cells. So this is what their design is, and biomarkers that they have tested is EGFR and PD1, and if they are present, then they should give this hybridization chain reactions in 13 minutes. And when this hybridization chain reaction occurs, they will see this fluorescence readout. And for negative control, they have used only EGFR biomarkers without any PD1. They have used only one biomarkers reagent without another reagent. And similarly, when they used PD1 without any EGFR, so they should not give any fluorescence signals in negative controls. And they tested this their biomarkers logic gate in A431 cell lines. So when they used only one biomarkers, that is in this case they have seen very low or faint image of this PD1, so they do not see fluorescence signals. When they use both the biomarkers, they are able to see fluorescence signals. And when they use only this EGFR, then also they see very minimum fluorescence signals. And this is for the 8 nucleotide. And for the 6 nucleotide, here they can see some non-specific signals also. And when they did it for the 6 nucleotide, the non-specific signals are very minimum, and the specific signals intensities can clearly be distinguished from the other negative controls. And for 4 nucleotides, they are it do not give any signals even when both the biomarkers are present. So this is like too short, whereas 8 is too long, and 6 seems to be the appropriate for the AND logic gate. And for that they tested one more biomarkers. They tested one more biomarker, tested it with one more biomarkers. That is, instead of this Z EGFR and Z PD1, they changed one of them and checked whether it will still give some non-specific signals or not, but do not see any non-specific signals. So therefore they decided to move with this 6 nucleotide, and the quantifications are shown in the panel C, where they can see that the intensities of with this hybridization chain reactions, when only this one of the nucleotides are present, then it gives some amount of non-specific signals, whereas when we see it for the 6 nucleotide, the non-specific signals are very minimum compared to all three cases. For the 4 nucleotides, they do not see any fluorescence signals, high fluorescence signals, even when both of them are present when the hybridization chain reactions occurs. This indicates that the 6 nucleotides, this result shows that 6 nucleotide would be the appropriate candidate for this AND logic gate. So they took this 6 nucleotide AND logic gate system and tested them in different cell lines, and they targeted the cell lines that have both the biomarkers with single biomarkers and one that does not have any biomarkers. So with both biomarkers, that is A431 cell lines, they are able to see fluorescence signals. But in the case where they have only like one biomarker, that is U251, which has only PD1 bio presence, biomarker present, PD1 biomarker present, they do not see fluorescence signals, or fluorescence signals are very low. And similarly, the MB468 cell lines, which has this EGFR, but they have very minimum PD1, they see very minimum or almost negligible fluorescence signals. And the MCF7 that does not have any biomarkers, they do not see any fluorescence signals, and these things they are quantified in the panel E. So these results are also in line with their expectations. So this results, that is the efficient and logic gate system, they have tested it on different cell lines and get the expected results. So from that, at this point of time, they are able to make an logic gate system which has a very minimum leakage. At the same time, it gives the signals when both the biomarkers are present. So this is what they have presented. This is what they have presented in this figure. It is their AND logic gate optimizations. If you need to understand more about this experiments and their AND logic gate system in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.